today's webinar will be recorded and available later. I'd also like to draw your attention to the right side here. There's a chat window. So if you have a question during the webinar itself, please feel free to type that question into the chat box and we'll be sure to answer it at the end. I'd also like to take a minute to just recognize all the hard work in the research and understanding of this virus that those here on our teams and on the webinar and worldwide are doing. You know, we're at Swift, we're very thankful to be collaborating and joining this community. I think this is very important work. So thank you for joining us and participating in this with us. Go to the next slide, please. So introduce our speakers today. On the left here is Caleb Heck, who is our R&D scientist for the RNA workflows at Swift. And then on the right here is Brian from Arbor Biosciences, and he'll be speaking to you about some of the Hybridization Capture products from Arbor Biosciences. Next slide, please. So just as a brief introduction to our two companies here, Swift Biosciences has been in the library prep space for NGS library preps. And we have launched our Swift RNA and Rapid RNA library preps late last year. Seen a lot of traction in the market for a variety of RNA sequencing projects. And we can work directly from first-strand cDNA using our adaptase chemistry, shorter workflows and those which handle very low input material. And then Arbor Biosciences specializes in target enrichment using their myBaits technology. So we're very pleased to be collaborating here to offer our respective expertise in these two workflows to bring a workflow for targeted sequencing of the coronavirus. So with that, I'll hand it over to Kayla Peck and our team, and she'll introduce to you the work that she's done along with Arbor Bio to show this workflow.
All right, thanks, Drew, for the introduction, and thank you all for being here today. Before we get started, I'll just go ahead and give a little bit of background just for why we're all here, which is coming up with ways and workflows to accommodate all of the new SARS-CoV-2 research that's happening. So SARS-CoV-2 is a single-stranded RNA virus that has a genome size of about 30 kb. I have that depicted here. You can see that there's a variety of different genes. The virus is present in a virion down here on the bottom left. And you can see that the genomic RNA for that virus is packaged within this capsid. And the genome is normally surrounded also by, or bound to nucleocapsid protein. Coronaviruses have a lot of different interesting ways in their, that they do the replication. One has this subgenomic mRNA. Sorry, hang on a second. I think there's some feedback that's happening. Just 1 minute.
Yeah, we'll handle that, Kayla. If you just might want to close your Teams app there.
People are saying that there's no sound.
We can hear you on the GoToMeeting, Kayla. Recommend just shutting down your Teams app.
All right, yeah. Sorry about that, everybody. Okay, so coming back to the SARS-CoV-2 genome, the current methods that are in place for diagnostic testing normally revolve around using a qPCR assay in order to target a very small region of the genome and see whether or not it can be amplified. Because it's an RNA genome, the first step would be reverse transcription to convert it to cDNA, and then that cDNA is used as the input for the downstream assays. For example, the CDC test that was in place had primer pairs that targeted the nucleocapsid gene in order to see whether or not that RNA piece could be amplified. While the qPCR assays are a great way to rapidly determine whether or not this RNA is present, there's obviously going to be interest in being able to sequence the full-length genome. RNA viruses mutate extremely rapidly, and so the ability to be able to track the mutations and nucleotide variants that are occurring in the SARS-CoV-2 population is going to be extremely informative. Not only can it help in epidemiology studies and help understand how the virus is transmitting across the world, it can also help in terms of pathogenesis studies and trying to form relationships between certain mutations or certain variants and the outcome of patients. And so obviously the the field that is going to be sequencing the full-length genome is going to be an extremely important one. But unfortunately, there are a lot of challenges that come with trying to sequence the SARS-CoV-2 genome. Some of these occur in the fact that there's going to be extremely variable sample types, so how those samples are collected, where they're collected from, and there's also going to be variation in sample quality. So in terms of when a swab was taken, for example, and the time before it's actually processed. RNA itself is much less stable than DNA, and so there's a lot of different components that can lead to a reduced sample quality. There's also going to be low viral titers in most of the samples that are collected. With low viral titers, that means there's less RNA present, and that means that we have to have methods that are able to detect a very limited amount of that starting material. There's also going to be variability in methods for RNA extraction, and then there's also the issue of when you extract that RNA, it is difficult to separate out the viral RNA from the host RNA, and so any sample that you have will be heavily contaminated by the host genomic RNA. And so these are all of the challenges that we have to face when we're interested in being able to sequence the entire length of that SARS-CoV-2 genome. The workflow options that you can imagine come in two forms. One would be one that is not targeted. And so this could include, for example, taking your RNA sample and then putting it through a ribodepletion module and then going into an RNA-seq library prep. This is because, as I mentioned, most of the RNA is going to be that host genomic RNA. And we also know that ribosomal RNA makes up 80 to 90% of an RNA sample. So by depleting that 80 to 90% of the ribosomal RNA, you're able to increase the proportion of your viral RNA within that sample. You can then sequence that sample, and a lot of your sequencing reads are going to be the host genomic RNA, but you would be able to get some of the viral RNA as well. The other option is to take a targeted approach. So for example, this would be taking your sample and going into an RNA-seq library prep, and then taking that library and putting it through hybridization capture. Hybridization capture is essentially a workflow that allows you to use designed probes to target just your library molecules of interest. So even though you create a library prep from your total RNA sample, you would then be able to enrich for just the components of that library that you're interested in. And this is the workflow that we're presenting today. The RNA-seq library prep comes from Swift Biosciences with our Swift RNA library prep kit. And then the hybridization capture is coming from Arbor Biosciences with their myBaits panel. And I will just note that there is another targeted option. For example, if you produce cDNA and then went into an amplicon panel, and that is something that Swift Biosciences has in development, but it is not the workflow that we're focusing on in the webinar today. So if you compare a not targeted versus a targeted workflow, if you're not targeting the specific SARS-CoV-2 sequences, you can imagine that this becomes very costly in terms of sequencing. The majority of the reads that you get are going to be to your host RNA and not to the viral RNA. Additionally, because you're not enriching specifically for viral RNA, it would require samples that already have high viral titers, which aren't necessarily going to be the predominant samples. However, with targeted, because you're enriching and sequencing only those molecules of interest, it's much more cost effective. And because you're amplifying and enriching for the SARS-CoV-2 genome specifically, you can see that there's a lot of different RNA molecules and species within this. Most of them are going to be your host RNA. So the purple would be your mRNA transcripts. The blue would be that ribosomal RNA, which again, we know takes up the predominant amount of that sample. And then you'll have a little bit of the SARS-CoV-2 RNA within that sample. You take that entire mixed sample and you go directly into the Swift RNA library prep. What you get out of the Swift RNA kit would be a fully functional library. So what we have here are diagrams of what a library molecule would look like. So this center block here, for example, in this top one with the green, that would be your insert. So this came directly from your RNA sample. That's the sequence corresponding to SARS-CoV-2 in this example. And then the color blocks on the end are representing the adapters that must be added in order to make a molecule compatible to go on to an Illumina instrument. So these molecules are already fully functional library molecules that can be sequenced. But in this workflow, we would not sequence this directly because, again, our coronavirus genome is a very minimal proportion of our full sample. Instead, you have your capture probes that have been designed to target the SARS-CoV-2 genome specifically through hybridization capture, and then you can enrich for just those molecules. So what you'll actually end up sequencing are your library molecules that were produced from the Swift RNA kit and then enriched and selected for from the Arbor Bio Hybridization Capture panel. I'll go over the workflow of the Swift RNA kit just so we can get an idea of what that workflow looks like. As Drew mentioned, we launched 2 RNA kits last year. One is the Swift RNA library kit, which I'll focus on here, and the other is the Swift Rapid RNA kit. Both of our RNA kits are powered by our adaptase technology. Our adaptase technology also kits such as our methyl-seq kit and our 1S single-stranded DNA kit. But the adaptase technology actually has several really useful functions in the context of our RNA-seq library kits. And I'll go over those after I detail the workflow. So the first step is you have your RNA, and then you use a random primer to undergo reverse transcription to produce first-strand cDNA. Now, because we have adaptase technology, and I didn't define what it does. Adaptase acts by allowing you to add an adapter directly to a single-stranded molecule. So in this context, this is really beneficial for RNA because it allows us to directly add this adapter to the first-strand cDNA molecule. Most other RNA-seq library kits require you to produce the second-strand cDNA before you're able to add any adapters. So already we've saved time by allowing us to add an adapter directly to first-strand cDNA instead of requiring that lengthy second-strand cDNA synthesis process. We then have an extension step and a ligation step to get the other adapter onto that molecule. And then we finish it with an indexing PCR step, which adds your barcodes and also creates your full-length adapters to make it an Illumina compatible library. Now, in this workflow, adaptase is doing several things. I already mentioned that it saves us time by allowing us to skip that second-strand cDNA synthesis step, but it also allows our library prep kit to be stranded. Because we know that this adapter is always getting added to the 3' ends of our cDNA molecules, we always know the orientation of that starting fragment. Second, it's also allowing us to separate our adapters in space and time. This results in very minimal adapter dimers. So because we have cleanups between when the first adapter is added to the reaction versus the second adapter down here in ligation, we have very, very few adapter dimers forming. And this is important because a lot of other library prep kits have the 2 adapters in the reaction at the same time, which result in lots of adapter dimers forming. And because of this, those kits also require you to titrate your adapter based on your initial starting input. But adaptase allows us to treat all samples the same regardless of their starting input, which simplifies the process. I have a table with some for our Swift RNA kit. And so in a traditional workflow, if you're starting with a mammalian total RNA sample, you would want to go into some sort of upstream module like poly selection or ribodepletion. In that sense, the Swift RNA kit can support inputs down to 10 nanograms. So that 10 nanograms would be your amount of RNA going into that upstream module prior to going into the Swift RNA kit. However, in this SARS-CoV-2 workflow, we are taking total RNA and going directly into the Swift RNA kit without any processing upstream. So in that sense, the Swift RNA kit can go down to 100 picograms of input. And this is actually one of the reasons why we're focusing on the Swift RNA kit. Again, I mentioned that we have a Swift Rapid RNA kit, and that is an expedited workflow. This workflow takes 4.5 hours, which is quite fast, but the Rapid kit is even faster. However, because of that expedited workflow, it can't support as low of inputs. We know that a lot of these samples are likely going to be limited in the RNA that's able to be extracted. And so Swift RNA is beneficial in that it can support and make library from limited starting material. We also, at Swift, have a variety of different indexing options that are available. And so we have enough indexing capabilities such that you can multiplex up to 768 libraries. So this will become important later when we start to show what sort of sequencing data you can get and read depth based on the amount of samples that have been multiplexed. So the data that Brian and I are going to show you today starts with taking the SARS-CoV-2 synthetic control that's available from Twist Biosciences. So what this control is it's single-stranded RNA, but it's been cut into 6 different 5 kilobase fragments. And what this means is that because the genome has been cut up, we're able to work with it at biosafety level 1. So it's still the full-length genome, it's just been cut up into pieces. It's also present at a known viral copy concentration. And so what we were able to do is we could take different numbers of viral genome copies from this control and mix it into a total RNA sample. We used universal human reference RNA. It's a traditional reference RNA sample that's very diverse. It's made up of 10 different cell lines. And so this is to mimic a sample that might be extracted from a swab, where you would have a lot of that genomic host RNA and then a very little amount of your actual SARS-CoV-2 RNA present in that sample. We tested between 0 and 1 million viral genome copies mixed in with 10 or 15 nanograms of the universal human reference RNA. So this entire mixed sample would then get put directly into the Swift RNA library prep kit. What you get out of that kit then is a functional library that has molecules that are made up from this entire mixed population. And I will note that we, in the Swift RNA protocol, we do have protocol recommendations for how to adjust your workflow when you know that your samples are going to be going into hybridization capture downstream. So since we knew that our application was hybridization capture, we followed all of those recommendations in order to adjust the protocol slightly to best fit with a hybridization capture downstream workflow. And so because what you get out of the Swift RNA kit is a functional library, I decided that we would sequence that to see what we would actually get. So we already know that we expect to have a lot of the host genomic RNA present in that sample, but what would it look like if you actually just sequenced it as it was? And so that's what these results are showing here. What I have is I have the 1 million copies of the SARS-CoV-2 genome spiked into either 10 or 50 nanograms of that universal human reference RNA, and they were sequenced to a depth of about 7 to 10 million reads. And what you're seeing is that there's a very limited amount of SARS-CoV-2 reads that are coming out of this data. So for our 50 nanogram sample here on the bottom, we have 0.01% of our reads in that SARS-CoV-2 genome. If you look at these IGV plots down at the bottom, I'm showing what that coverage looks like. And so even though we're seeing, you can see that with all of this color, it's very noisy. So even the coverage we're seeing, we're not actually very confident in. And so this is really just showcasing why we need something like a hybridization capture or some sort of enrichment, because not only do we not have high confidence in this data, it's very wasteful when we've just wasted almost 7 million reads to not get what we want. And so with that, I'll go ahead and hand it over to Brian so he can talk through the hybridization capture of this workflow.
All right, thank you, Kayla. Can everyone hear me okay?
Yeah, I can hear you fine.
All right, great. Thank you again for the intro. And as Kayla mentioned, this is where enrichment of low abundant targets in a sample using hybridization capture is a really effective solution. And just to review the workflow for Hyb capture that Kayla described, it's a straightforward and user-friendly procedure with no special equipment required. Essentially, a barcoded NGS library or pool of libraries is denatured and allowed to hybridize over a period of hours to the complementary single-stranded RNA biotinylated synthetic probes, or baits as we call them. After hybridization, the probe and target molecules are bound to the streptavidin-coated magnetic beads and washed with buffers to remove non-target or loosely bound library molecules. The target library molecules are then released and amplified with the universal primers to increase not only the library concentration, but also to restore their double-stranded format so they can be sequenced. Arbor Biosciences specializes in designing and manufacturing customized Hive capture kits under our myBaits product, and we have extensive experience with specialized applications like pathogen sequencing for unique targets like viruses and bacteria. Next slide, please. Therefore, to meet the need within the research community for the full genome sequencing of the novel coronavirus SARS-CoV-2 genome, we designed and produced a set of comprehensive SARS-CoV-2 target capture probes and kit. Our design incorporates all full and partial genome sequences that were publicly available on NCBI as of January 31st of this year. Overall, the design has over 2,000 overlapping 8-mer probes with an effective tiling density of 6x. So that means there's about 6 probes per nucleotide in the 30 kb genome sequence. After filtering to remove low complexity probes, so that have simple repeats like just AT, 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 as well as probes that could cross hybridize to a variety of potential host genomes and like humans and animals that would bias to capture. About 99.9% of the genome is represented in this comprehensive design. Please note we've continued to monitor the CoV-2 genome content over the last few months, and this design is still fully functional to capture all the novel variation. And this is due to the very flexible power of the Hive capture system to accommodate a significant amount of mismatches between probe and target molecules and still work very effectively. Next slide, please. The HiveCap procedure falls conveniently between the upstream creation of an index library and the downstream NGS sequencing, which makes it just really one extra step in the normal pipeline. And again, because the libraries are already barcoded at the point of capture, this step can and is often performed on a pool of libraries for maximum convenience, flexibility, and cost effectiveness. Typically about 4 to 12 libraries can be pulled per capture, but this really depends on your target and the sample type. If you're working with really degraded DNA, you might want to pool fewer samples. All right, next slide, please. And so looking back at what Kayla presented, our experiments using the Swift RNA libraries containing the low level of the SARS-CoV-2 control fragments demonstrated highly successful enrichment. As you can see in the table, the Hive enrichment was able to recover reads comprehensively spanning the entire viral genome and increase the percentage of total viral target reads by orders of magnitude from like under 0.1% to more than 80%. Full genome sequencing is therefore possible with only a fraction of the total read depth that would have been required via direct sequencing of the libraries. And please note, these are done on the same exact libraries. The sequencing is done on the same exact libraries before and after enrichment. Next slide, please. And so this graph demonstrates the pattern of read coverage for the same set of libraries pre-enrichment, which are in purple, and post-enrichment, which are in green. And we observed a very similar overall pattern of read coverage, with essentially very few regions having higher, lower than needed or expected coverage depth than either group. You look at the x-axis, the normalized coverage is between, is very tight around 1 with not many outliers. And overall, this demonstrates the ability of Hive capture to preserve the relative target read abundance patterns of the original libraries. So if you wanted to know relative abundance of this sample versus another sample, you might be able to determine that from the same sample. Next slide, please. All right, this graph shows the total percent of reads aligning to the viral genome, which is the y-axis, for libraries with 10, 1,000, or 1 million copies of the viral genome, which is the x-axis, in either the 10 or 50 nanogram total human RNA backgrounds. And this is done at a total sequencing depth of 250,000 reads. And we get very reliable detection of the CoV-2 genome, even down to 1,000 total copies of the viral genome. And when we look at the million copies, viral copies, which keep in mind is still less than 0.1% of the total library that was inputted, we're able to achieve the vast majority of reads, about 80% of all the reads mapped to the viral genome after enrichment. Next slide, please. And now if we look at the fold enrichment, which is the y-axis, we can see we are able to increase the total amount of viral genomic reads by orders of magnitude with the myBaits Hive capture. And by more than 1,000-fold in the very low 1,000 copy libraries and nearly 100,000-fold enrichment in the 1 million copy libraries. Next slide, please. So if we examine the percent viral genome covered, which is the y-axis, we find that even as low as 1,000 viral genome copies, we're able to recover reads mapping to a significant portion of the genome. As the graph shows, with 250,000 total read depth, more than 8% of the genome is at least 1x covered, and more than 50% is at least 2x covered for the 100,000 copy target. For the million viral genome copy, which again is still less than 0.1% of the library, the entire genome is easily fully recovered at this total sequencing depth, with the majority of the library's reads now mapping to the viral genome. And this facilitates the pooling of samples, as I talked about, for sequencing together on like one sequencing lane. Next slide, please. So the next question is, what sequencing depth is needed? And this will depend, of course, on the goals that you have for your study. But even with samples where the target viral RNA makes up less than the 0.1%, the full genome resequencing with high read coverage can be achieved with less than 100,000 total sequencing reads. And for example, with only 65,000 reads per sample, one could easily pool together 384 samples on a single MySeq run and still achieve full genome sequencing with at least 10x coverage. If you wanted greater sequencing depth, for example, 250,000 reads, one can still pool at least 96 samples on a single MySeq run. Next slide, please. And so hybridization capture is a powerful tool for flexible targeted next generation sequencing, allows you to harness the full power of your chosen NGS platform. It's highly tolerant to accommodate any type of sequence mutation types, such as SNPs, indels, and rearrangements. And the tolerance of significant diversity, including novel synteny, makes it the ideal technology approach for pathogen targeted sequencing, like in viruses, where unknown samples can be significantly different versus a known reference genome. The same flexibility of the probes also allows Hyb capture to excel even for very short or degraded target molecules. And this is where Kayla was mentioning before, where qPCR works, but if you have a breakpoint in there, it may not amplify, whereas the probes would still be able to bind to these small fragments. And in this case, as always, one critical step for the Hyb capture is to make sure you have high quality libraries like those made by Swift for containing a sufficient abundance of target molecules. Next slide, please. So with hybridization capture, it is easy to mix and match different sets of probes for customized target sets, making it ideal for pathogen sequencing research programs across a wide variety of applications, such as genomics, evolutionary biology. Like you could actually combine our SARS-CoV-2 kit with a custom kit if you wanted to expand it and look for multiple different viruses within the same sample. The targets can be pathogen with either an RNA or DNA genome, again, as long as a good quality NGS library can be made from the sample that contains the target molecules of interest. Arbor Bio is working on expanding our line of pre-designed target viral panels, including a pan-coronavirus panel, and of course more. Please contact us to get our full list of future updates or to get started on myBaits custom pathogen kit that we can work on immediately for viruses, bacteria, or whatever your target of interest would be. Thank you.
All right, thank you so much, Brian. So just to summarize, this collaboration between Swift Biosciences and Arbor Biosciences has allowed us to take the Swift RNA library prep kit and produce high quality RNA-seq library that can then be used with the Arbor Biosciences myBaits SARS-CoV-2 panel. This is really critical because it's important to be able to sequence the full length of the SARS coronavirus 2 genome in order to be able to track mutation. But there are a lot of challenges in terms of getting a quality sample that can be sequenced. And of course, the host genomic RNA is going to take up a very high amount of that sample. So doing something like enrichment is really crucial for being able to get high quality sequence data for the SARS-CoV-2 genome specifically. We showed today that this workflow is really robust. It does not show any bias that's introduced by the capture. We also showed that we were able to detect and have good data down to 1,000 viral genome copies, even when it makes up a very, very small percentage of the overall RNA that was input into the library. We had enrichment rates up to 90,000 fold, showing the power of being able to capture and enrich for just those target molecules of interest. And that enrichment led to over 99% of the SARS-CoV-2 genome being covered. So really allowing you to detect those mutations and have confidence in the variants that you're calling. We showed that if you have a sample that has even 1 million copies, which is still a very low amount. You can have over 100x coverage with just 100,000 paired end reads. And this really is showing you the ability to be able to multiplex and add samples together and still have really great coverage of your SARS-CoV-2 genome for whatever application that you have downstream. Here I have some information on how to order these components. And so again, the Swift RNA library kit would be your first processing library kit. And then we also have those variety of different indexing options. So as we mentioned, you can go up to 768 different combinations depending on your sequencing instrument and how many samples you wanted to multiplex. We have the Arbor Bio reagents here down here as well. And I just wanted to point out that we have these reaction sizes here. But as Brian mentioned, depending on your application, you can actually pool multiple libraries together to go into one hybridization capture reaction. So for all of the data that we presented, we pooled 4 libraries together in order to go through one hyb capture. So the 16 reactions would actually end up being 64 different libraries or samples that were processed. And so if you're interested in ordering these, you can reach out to your local sales rep from either Swift Biosciences or Arbor Biosciences, and they'll be able to get a quote and get you all the components that you need to go through this workflow from start to finish. And then I just wanted to note that we've actually been working hard on an application note that will have all of the details of the data that was presented during this webinar, as well as all of the information that you would need to adjust your methods and your workflow to be able to go from your RNA sample through the Swift library prep, through the Arbor Bio Hyb capture panel to your final sequencing on an Illumina instrument. And so this will be out by the end of the month, and hopefully will be a great resource for you to be able to maximize your data output for your SARS-CoV-2 samples. And with that, I just want to say thank you so much for joining, and Brian and I are happy to answer any questions that you might have on this workflow.
Thank you, Kayla and Brian. And we did have one question come in through the chat box just asking if we have had a chance to benchmark against other Hyb capture workflows for COVID. And feel free to chime in or add to this, but you know, we haven't had that opportunity yet. We've you know, been focusing on getting this combined workflow out as quickly as possible, and so that's been the emphasis we've had here to date.
Yeah, that's correct. We haven't had a chance to benchmark, but we actually have used some similar conditions in terms of the amount of UHR and the spike-in for the SARS-CoV-2 for comparison with the Twist Biosciences panel. And so if you compare the data head to head, obviously it's not performed in the same lab, but we actually do see better coverage of the genome at lower sequencing distances.
I think the Swift library is a little faster too than what they recommend about. So our Swift RNA, right, Kayla, is about 4.5 hours, and the Twist I think is six plus hours to do just the library prep component. So that's another difference, as well as the price. You'll see a price difference there for sure.
And I'd just like to point out too, for the time being, Arbor Biosciences is offering one free kit to anyone actively working on CoV-2 research. So you can see the link at our website, and you can get one kit for free if you wanted to test it out to see if it worked for your needs.
Yeah, if you had any other questions, feel free to put them in chat or unmute yourself and ask.
This is Tom Fabrizio from St. Jude Children's Research Hospital. I'm curious, have you been able to test this on an actual clinical sample?
Hi, Tom. I think we've been in contact. Hope you're doing well.
Yeah, I am. How about yourself?
Doing good. We haven't for lots of reasons, mostly biosafety levels, but we've had discussions with the customers, and of course I can't reveal their data, but we've many people who have worked on clinical isolates and have been very happy with results. of course, they will share their results when they are ready, but been very positive feedback, and people have ordered more kits.
Sounds good. Thank you very much.
Thank you.
We have another question from the chat box, whether we recommend ribosomal depletion or if it would improve the results, as well as if we have any experiments with real host material contaminating the sample.
Yeah, so for ribosomal depletion, I've performed experiments doing kind of that non-targeted workflow where you do ribosomal depletion with the library and then sequence. And you really only see a 10-fold increase in the amount of data that you get. So previously, when you had 0.01% of your reads coming back SARS-CoV-2, the ribodepletion made it 0.1%. We haven't actually done ribodepletion into a library into a high capture. And it's my intuition, though, that because of how many library molecules you're producing before you go into high capture, that depleting before might not actually have that big of an impact. And so by adding the extra 2 hours of ribodepletion, you have a very minimal impact on your final library. Is a trade-off, but it's not something we have tested. But it's my intuition that it might not improve that much. And sorry, Drew, I didn't actually catch the second half of the question.
Yeah, the other question was just whether we've had a chance to test this with real host contamination or RNA from the host, the potential host.
Yeah, so we haven't actually, because we've been trying to do this in a more controlled setting, we haven't actually obtained RNA that would be the RNA that would be extracted from one of these samples. But that's definitely something that we would be interested in getting the data for.
Yeah, and perhaps this is to your point, Kayla, about the ribodepletion, but I don't know if you wanted to flip back to that slide showing the percentage alignment after Hype capture, and that would be the metric where you would see, is that correct? So you're achieving 97% or 87 to 97% mapping to the SARS genome without ribodepletion. And so you'd not expect that to change much with the ribodepletion.
Yeah, sorry, Drew, you were breaking up a little bit. But yeah, based on how minimal the ribodepletion impacted this metric, I just wouldn't anticipate it would have that big of an impact. You have a sample that you're already heavily selecting the SARS-CoV-2 molecules out of.
Okay, thanks. And we have another question from the audience. What would be the advantage of this approach versus an amplicon approach?
Well,
and
amplicon. Go ahead. I'd say amplicons have the benefit of speed, though you still have to sequence. Though the hybridization capture doesn't have any of the limitations of amplicons. One being that you don't have, if there's degraded samples or breakpoints that would be between two primers, are not applicable to the hybridization capture because it's a probe that binds to a region, whether it's 300 nucleotides or 60 nucleotides. Also, there's no bias from the different amplicon during the PCR step, as the, I forget which slide number it was, slide number 18. It shows that the coverage of the pre-enrichment and post-enrichment is the same. So you would retain any differences that are relative abundance differences using the capture probes. And this can be an advantage, like we have a 16S panel and allows you to faithfully recreate the shotgun metagenomic data as well for the 16S specimens. So those are the two big keys, is the lack of PCR bias, and you can use any type of sample as an input.
Thanks, Brian. And there's a follow-up question just confirming, with the Swift Amplicon panel, that is an alternative workflow. You're right, Patricia. It does have more than 300 overlapping amplicons to achieve similar coverage. But I think Brian's point is a good one, that if you're working with very degraded samples, the Hype capture is potentially going to pull down and capture those highly fragmented samples. One kind of point we'd like to make here is just that we are enabling both a Hype capture based workflow and an amplicon based workflow. We've had a lot of interest from the research community to make both types of tools available. And so we do see people testing both methods and looking at the results there. But it's both providing excellent NGS coverage and data. Okay, we'll give it a couple more minutes here. We really appreciate everyone's attendance and the active Q&A session. I did want to mention too that we are planning, in addition to the application note that Kayla mentioned, we are planning to have a couple of follow-up Q&A sessions over the next week. And so be looking at your email for those dates and times, and we'll be happy to continue the dialogue with anyone who'd like to join those over this webinar type format as well. But if there are any other last minute questions, please feel free to ask now. And otherwise, we'll be circling back with you about the application note, ordering information, and those follow-up sessions. Okay, with that, I think we'll end the webinar for today. Once again, to those that are still on the line, please look out for the follow-up information, and we'd like to really thank you for joining us today. Have a great rest of your day. Bye-bye.
Yeah, thank you, everyone. We appreciate it.
Thank you very much.
Thank you. Thanks, guys.